All right. Hello, everyone. My name is Inte, and today I want to talk to you about a tool that we've developed in order to infer regulatory dynamics, and then we applied that to breast cancer. Let me just get this. Okay. So here's the big picture. We are interested in gene expression dynamics. In other words, we're interested in knowing how a network of genes, how their gene expression evolves over time. And you can kind of think of the utility in two ways. First of all, it gives us an understanding of the nature of this biological system in question. And secondly, if we have an accurate understanding of the gene expression dynamics, we can predict the responses to interventions or perturbations. To be a bit more formal, the problem is, given time series gene expression data, let's say you have n genes, we want to estimate an n-dimensional vector field. So you want to get n different differential equations, one differential equation for each gene, where each differential equation for each gene depends on all the other genes. Now, I understand that time series gene expression data might not always be available for every kind of data modality, but we've built this tool to be kind of agnostic to the kind of data type. You can have microarray data, which is actually where you have time-labeled samples, or you can have single-cell RNA-seq, where the time is imputed based on some kind of a pseudo-time measure. Right? Okay, so we start with this tool that came out in NeuralODEs in 2018, they're called the NeuralODEs differential equations, which aims to estimate the local derivative. Given a vector of gene expressions, you aim to estimate the local derivative at every time point using a neural network block. This local derivative is then integrated through an ODE solver to give you the next time point. And so this backpropagation needs to go through this ODE solver and then through your NeuralODE and through your neural network. Therefore, this neural network parameterizes the derivative, and it's exactly what you want. The problem with this is that if you've used NeuralODEs in any of your work, you'll know that they're kind of hard to work with. And they are also not very scalable and quite black box. So an out-of-the-box NeuralODE is not really biologically explainable, and as a matter of fact, is not very scalable. So if you have something more than 10,000 dimensions, and my apologies for the typo there, more than 10,000 dimensions or 10,000 genes, then this starts to, it's not very scalable. It starts to give very poor performance. And I want to note that this is also a problem with a lot of the other methods that are currently being used. You might be familiar with some of them, but a lot of them simply cannot scale to a large number of genes without using some kind of a dimension reduction step in between, in which case the sort of biological explainability or interpretability is lost. Now, we solve this problem by doing two things. The first one is we come up with a biologically motivated neural network architecture that is based on Hill kinetics from biochemistry. Specifically, we have our neural network block over here, you'll see is modified, where we have separate parts of the neural network trying to model both additive as well as multiplicative effects. We also have activation functions, which are inspired by Hill kinetics, so as to give it a more biologically kind of inspired flavor. The next thing is this addition over here, where we try to make it more biologically explainable. So we have some kind of a prior domain knowledge model, and this can be any kind of prior knowledge model or prior constraints, which the model tries to simultaneously fit along with fitting your data. In the process, what happens is that this neural network explains the evolution of gene expression over time in a way that respects the constraint of your prior model. Now, a natural question could be, what are these prior domain knowledge models? Now, we've built it to be extremely flexible. These prior domain knowledge models can be any kind of biochemistry-inspired complicated differential equation model that you like. They can also be something as simple as just a network of putative transcription factor to gene interactions. They can also be a spatially informed transcription factor activity network. It can also be something empirical. So if you have empirical data mapping expression to RNA velocity, you can also do that. So essentially, we have a neural network that gives you these derivatives or these ODEs that we integrate to get the next time point, and we enhance this process with explainability by using these prior domain knowledge models. Our model is called Phoenix. This is quite a tortured abbreviation, and if you don't like this abbreviation, and neither do I, but it's the one we have, right? Okay, so in the next few slides, I have 4 more slides, and I want to show you 4 results or 4 advantages of a modeling approach like this. The first one is that Phoenix is quite predictive, and it's predictive out of sample. The way we do this is we have in silico experiments starting from some ground truth gene regulatory network, and using the connectivities of these ground truth networks, we simulate time series data. We benchmark Phoenix against a bunch of other methods in the field, and some of these methods require additional data inputs. Because we're using a in silico or we're using simulated data, we actually feed into these, as these additional inputs into these other models, we feed in the ground truth information itself, thereby mimicking kind of the best possible performance of these competing methods. We also tune their hyperparameters using a cross-validation approach. So we see that Phoenix outperforms these methods and kind of outperforms optimistic versions of these methods, right? Because we've also optimized them. The second thing is explainability. So as I mentioned, like part of the architecture is trying to imbue this prior knowledge or prior information, and therefore we really care about explainability. We noticed that the Phoenix model is actually quite simple, and therefore the parameters of a Phoenix model can be used to infer some kind of a causal gene regulatory network that we can compare back to the gene regulatory networks used to generate the data in the first place, right? And we get something like this. If you've ever played FIFA, these will probably be very familiar to you. So we're essentially comparing, these are various different like graph comparison metrics looking at explainability. So we have area under the curve, true positive rate, true negative rate, some measure of sparsity. The first thing is to see this blue polygon, which is Phoenix, and we compare it to an out-of-the-box NeuralODE. And we see that we completely dominate an out-of-the-box NeuralODE in terms of explainability. The red one is the Phoenix model where we gave 0 weight to the prior knowledge. And you'll see that this kind of quantifies the marginal benefit of including prior knowledge in the model. And as we get more and more noisy, Phoenix remains robustly explainable. Last two things, where we applied it to real data. So my apologies, I think, yeah, this one. We applied this to about a 3,500 dimensional vectors, 3,500 dimensional gene expression time series data from yeast, where this prior model was based on a motif map of promoter targets, and we also validated explainability based on ChIP-seq data. I know this is not a cancer example, but my point here is that even though Phoenix is based on hill-like kinetics, there is nothing sinusoidal about it, the model is able to deviate from hill kinetics assumption when needed and model arbitrary sinusoidal dynamics as per the data's requirements. It's still quite predictive and explainable. Lastly, we applied it to breast cancer data on about 11,000 dimensions. And these are breast cancer microarray data across 186 patients that we ordered in pseudotime. So we got this pseudo trajectory of breast cancer expression. We see that if you look at smaller subsets of the genes and then slowly scale up, Phoenix tends to stay explainable as well as predictive. And finally, this part in bold is, to our knowledge, the first time that this has been done on such a big scale without dimensionality reduction. So this is an 11,000 dimensional vector space, not using anything like a PCA or a UMAP. So you get actually these interpretable differential equations there. The last thing you can do with this is once you have this full like vector space, right? This scaling allows the discovery of candidate genes and pathways, which can be novel drivers of breast cancer progression, or novel Ryan Goslings, if you will. So the way we do this is we take a trained Phoenix model and we do small perturbations to see what are the genes that are actually driving the dynamics. And what you see is, as we scale up, we discover obviously more and more driver genes, and specifically driver genes that you do not catch if you're looking at a smaller subset. We can translate these scores into pathway importance scores as well, and that shows you that there are certain pathways that you're able to capture only when you're able to scale up. That brings me to the end of my talk. I do have a preprint if you want to take a closer look, and I have a poster in the main session, in the main conference, and I thank you for your patience. Thank you.
We have time for one question.
I guess if I get one question on it, then can you use causal graphs, like can you use causal discovery, and then move back and put causal graph, putative causal graphs in as constraints?
Yeah, absolutely. Yeah, exactly. Yeah. So you could have, I see what you're saying. You could have a causal graph, and you could put that in here as your prior model as well. It's quite flexible. Yeah.
So okay, well, if it's a very fast question,
Yes,
Yeah.
just in your prior model, can you also include like conditional, like categorical choices? Like you don't know if it's one or another model, like two alternatives.
Yeah, absolutely. Yeah, this prior model can also be condition specific. I obviously glossed over a lot of mathematical details. I'm happy to talk to you after this about how this can be done.
Right.
Yeah.
Okay, so let's thank all the speakers.